Hello, everyone. Thank you for attending my presentation. My name is JD, and I'm from Nemes Research Laboratory at the University of Maryland. My topic is about protein metabolomic single-cell system biology by using microsampling and CMS. System biology is important. It helps people to understand a large big picture of biological systems, and it has many applications such as studying metabolic network and cell heterogeneity. To perform system biology, people need to measure metabolite proteins and other biomolecules. However, the current single-cell analysis only focuses on either proteomics or metabolomics. Here is a few examples of single-cell analysis. There is no study for both metabolomics and proteomics analysis of the same cell. So our motivation is to perform, for the first time, metabolomics and proteomics single-cell system biology. To do that, we need a proper sampling technique. There are several available single-cell techniques, such as dissection and dissociation. The left picture shows dissection. People use it to isolate embryonic cell. The one on the right side is people use dissociation to isolate target cell for single cell analysis. However, this technique have limitations. They lack the space and the time scalability. Also, generally these two sampling techniques cannot maintain the integrate of the sample organism. However, these are important capability when we work with developing embryo. So here is the video of a developing embryo. You can see that the cell can live very fast and the position keeps changing. Dissection and dissociation, because of the limitation, they cannot be used working with this system. Therefore, new sampling tools is needed. Microsampling can be used to address the technical gap. It's scalable. During microsampling, a capillary is used to aspirate a cellular content directly. Here is the microsampling setup that we are using. Basically, we use a translational stage to control the capillary so that we can sample directly from the cell. Our goal here is to perform dual metabolomics and proteomics single-cell system biology using microsampling. And here is our proposed workflow to achieve our goal. It involves in vivo microsampling and single-cell mass spectrometry. This workflow divides into two parts. The first part is the bioanalytical chemistry, and the second part is developmental behavioral biology. In the first part, we use microsampling to aspirate from D1 cell trace. And one sample is used for proteomics analysis. The other sample is used for metabolomics analysis. And then we combine the data to do the multi-omics analysis so that we can use the pathway analysis and the statistical analysis. For the microsample embryo, we culture them and let it go through different development stages, such as the novel stage and the tadpole stage. And then once they reach to tadpole stage 4 to 5, we did the morphology analysis to score the organ. We also did the behavioral assay so that we can evaluate the visual function. This assay allow us to get the phenotype information. With the combination of the phenotype information and the multi-omics result, we are able to get a multi-dimensional data. This will allow us to do the molecular system biology. We are able to generate hypotheses for new for biological questions. We first evaluated the impact of microsampling on the embryo viability. We found that 95% of the microsample embryo survived to tadpole. And here are the images of the tadpole that are survived at tadpole stage. The left one is the control group. The one on the right side are from embryo undergo microsampling. By compare the organs such as green eyes, we found that these two groups are similar. There is no difference. That means the tadpole developed from microsample embryo didn't change the morphology. We then did the behavior assay. Specifically, we did the background color preference assay. This allows us to test the visual function and the swimming capability. Here is our setup. It consists of a camera and a tank. We place the tadpole into the tank and then use the camera to record the video. After that, we calculate the time percentage that a tadpole spent on the right side. And then we did the statistical analysis. We found that both the control group and the experimental group prefer white background. This matched with a previous study. Also, we found for these two groups, there is no significant difference. That means the tadpole developed from the microsample embryo and didn't change their behaviors. Now let's move to the chemical analysis. So here is the workflow that we use to perform protein metabolomic analysis of the live single cells. Here is our sample preparation workflow. We basically microsample the D1L cell twice. One is for proteomics analysis, another one is for the metabolomics analysis. Then we did the same thing for the V1 cell. After that, we use the HRMS to measure the proteins and the metabolites separately. In the HRMS analysis, we separate the compound by using capillary and then detect the compound using mass spectrometer. In our chemical analysis, we were able to detect 150 molecular features, which include 57 identified ones. In the proteomics analysis, we identified 738 proteins. We then combine the proteomics data and the metabolomics data to do the pathway analysis. The benefit is that we get the complementary result. So here is a few pathways that we revert. For the first one, you can see that it can only be reversed by the metabolomics data. For the glycolysis, we can only revert it by using the proteomics data. If we only perform one omics, then we will lose the pathway for some other. For pathway that detect the proteins and the metabolite, for example, the arginine proline metabolism, we are able to map both proteins and metabolite to expand the pathway coverage. Using the combined proteomics and the metabolomics data, we were also able to identify the cell-to-cell difference between V1 and D1. This allows us to discover the cell heterogeneity among early stage embryonic cells. So in conclusion, we enabled for the first time in vivo single-cell proteomics and metabolomics analysis in developing embryo. By performing this multi-omics analysis, we are able to reverse the cell-to-cell difference among early stage embryonic cell. And this difference cannot be detected in the single-cell transcriptomic analysis. Also, the results support our previous findings of metabolite-induced cell phase change. In the technical side, we provide a scalable technology that allow people to perform a complex biological system and different biological models. In the end, I want to thank my advisor for his support. I want to thank all the collaborators and the group members for their feedback. I want to thank the funding agents for their support. With that, I want to thank you for your attention, and I'm happy to take any questions. Thank you.